that we hear lecturing back to back is just a really remarkable, happy coincidence. And this is making me really excited. So I think Todd underestimated the value of embryoid bodies in terms of understanding the mechanisms of organogenesis. So in studying the embryo, or studying organogenesis in mouse embryos, this is just an aside before I start, able to follow organogenesis in the mouse live because you can only keep mouse embryos alive outside of the mother for about, anywhere somewhere between 8 and 12 hours. At really critical times when you want to find out about the basic mechanisms of, say, organogenesis in the kidney, in the heart, or the liver, or any of the multitude of other major organs in the body, you have a very narrow window in which you can remove those tissues from the embryo and actually study them live. Okay, so the embryoid bodies provide an opportunity and access to both the chemical environment of those forming organs as well as the mechanical environment of those forming organs. And so as an exploratory vehicle, I think they're tremendous. As an engineering vehicle, I think they're also going to be remarkably powerful. They may not be the technology that we use ultimately, but they will be a test bed that engineers can take and practice and test their hypotheses on. All right. So how is it that the embryo is actually grown? And I think the basic idea that I think of how embryos are built is that first you assemble the basic structure of like building a house. You build the foundation, you frame the walls, you put up the roof. And that's the very basic movements of early embryogenesis. Somewhat later, you call in the electrician, the plumber, the cable guy. You call in the phone company, and they string up all the sort of secondary things, all the organs that actually make the house habitable. Now, the difference between you building a house and the embryo building something like a house is that you would order all the parts at full size. In the embryo, the house is built about yay big, right, with all of the plumbing, everything else, and then you grow it up to the size of your 3-story house. So there's a tremendous amount of growth that takes place in the presence of functioning organs. Okay, and I think that's sort of the remarkable thing about this transition from an early embryo until later stages where you have a physiologically functioning organism. Okay, so what I'm going to tell you about today is all about this early embryogenesis and the mechanics that follow. And so I'm really fortunate to have followed Ray Keller's lectures, and so I can bask in the idea that you already understand a fair bit of early embryogenesis. And what I'm going to tell you about is some work that we and others are doing to try to measure the stiffness and force operating within the early embryos, and how these processes may be regulated on a mechanical scale. So I'm going to show you a few time-lapse movies, and you probably have seen many more, about how individual programs, sort of subroutines of cell migration, directed cell migration, branching morphogenesis actually build structures. So the first of these is the generation of what's called the lateral line in zebrafish. The lateral line is a sensory package that's distributed down the body of the zebrafish embryo and exists in the adult zebrafish. And it's a way for the embryo and the adult fish to recognize the presence of predators and thus escape from predators. So this is a key sensory system in the zebrafish. What happens and how it's built is almost as if you would build a sensory system around, a protective sensory system around your house. You would start at the brain with a package of cells, and then you would string those cells out in a line from the brain to the tail, and at regular intervals, this sort of string of connectivity drops sensory packages. And those sensory packages are seen here as these little nodules. Now the program of which this works involves directed migration of an epithelial cell type moving through a densely packed field of cells. So this is not like you're growing this in any way. It's actually moving, and in regular intervals, this slug here sort of kicks out a package of sensory cells. Okay, this takes about 8 hours, and if you look very closely, you can see how these individual cells are organized in their migratory pattern. Now, this migratory pattern depends on feedback systems of signaling between the substrate, the leading edge cells, the rest of the cells in the package, and then the cells in this sensory system that's being sort of produced as needed and kicked off the back of this migrating slug. You have another system of what's called branching morphogenesis. This is an example from the Konstantinan lab showing the growth of a branch network within an embryonic kidney. So you can barely see the outlines of the mesenchymal cells here. All that's labeled with GFP is the embryonic epithelium of the kidney. But you can see how each of these branches is formed, how the sort of iterative process of branching morphogenesis works, or at least how it proceeds. In our work, we're very interested in how the cellular mechanisms, like say at this scale, operate to drive the process of gastrulation at this scale, sort of bridging the scale of multicellular processes like actin contractility to the level of many thousands and thousands of cells that drive the sort of dramatic shape changes that I think Ray Keller was telling you about. Okay, so for this work, I think, so my primary appointment is as a bioengineer, but I have a secondary appointment and a long-standing commitment to the field of developmental biology. And so I like to contrast how tissue engineers and developmental biologists are different, but also what kind of things they might share in common. So I think there's a commonality to these two fields. So tissue engineers are interested in forward engineering, translational research, basically trying to apply fundamental laws to build artificial structures and ultimately to produce useful devices. Developmental biologists are not nearly so practical as that, right? So primarily they love to do sort of reverse engineering, puzzling out the pathways and the networks that drive early development. It's very basic research, but they're figuring out fundamental laws, like these fundamental laws of gene function, the establishment of pattern in the early embryo, and how, in my particular case, how structures are elaborated during morphogenesis. Now, this common region here, I think, is particularly interesting. So there are many shared interests, and the first of, there's many shared interests between bioengineers and developmental biologists, very practical ones like bioimaging and how to quantitate cell and molecular biology, features how to be very quantitative about these things. But I think we also share interests in the ability to reverse and forward engineer tissues and organs. And I think our goals, and Todd, I think, shares the same interests, is to understand how to turn modules of development into a new technology. So when we think about the major advances in engineering, we think about how electrical engineers build circuits today. They don't start with a random array of components, pick randomly out of the box, put them together into a circuit, and then build maybe 10,000 circuits and only keep the ones that work. Instead, they have simulation tools where they really understand how each of those components work, and they simulate that structure before they actually go and build it. We don't have that as tissue engineers, and I think the future really requires those kinds of both the simulation tools, but also the fundamental understanding of what are the different components. What do these very basic modules do? Okay. And so I think we can start to think, and this is going to be sort of my last grand theme slide, but I think we can start to understand, take this principle of control theory and begin to understand how it works on the scale of development. So at the level of genes, controlling protein expression, which then controls cell behaviors, which produce, if in open loop kind of configuration, morphogenetic movements. But if those movements go wrong, or if they're somehow outside of the normal parameters of a healthy developing embryo, there might be these control circuits. So there have to be ways in which which the output is measured and compared to some sort of input. And so this circuit is actually, I think it's sort of a crazy way to, maybe a crazy way to think about development, because we have no idea how output is compared to desired inputs. But I think it's a start to begin to think about these problems. Okay, so what I'm going to be focusing on here is the mechanics of development, the first lecture on mechanics of development, and I'm going to be focusing on very early movements, sort of basically building the foundation, the walls, and the roof of the embryo, setting up what's called the basic body plan. Okay, so and the question is, what is morphogenesis? And I think it's both the cellular and the mechanical process that shape tissues and organs. And then how is this process studied? So typically, and I think the first part here really encompasses all of developmental biology. So the idea is to manipulate and assess events within complex multicellular tissues, either through molecular perturbations, chemical perturbations. Now we're using physical or even optical perturbations. Now we're taking some approaches to measure the mechanical processes and properties in these embryonic tissues. I think there's a few groups around the world studying these processes in vertebrates. A fair number of groups are studying them in Drosophila, for instance. But I think I can demonstrate today that the model system that we're working with, and perhaps the embryoid bodies, provide significantly greater access to controlling some of these physical mechanical properties. And then there are systems approaches using computer simulation and synthetic biology to try to directly control morphogenesis in sort of a forward-thinking way. Okay, so why would we study? So first of all, this is sort of the part on your grant proposal that's, I think, critically important. To understand the basic science of morphogenesis is really going to help us understand the origins of birth defects and the environmental risk factors, the genotypic risk factors of birth defects, and perhaps suggest methods of prevention. Another aspect of morphogenesis is the problem of dysmorphogenesis during wound healing, and also during very chronic diseases such as cancer. The dysmorphogenesis is really a poorly understood feature of these diseases, but is often a critical contributor to the lethality of those diseases. Okay, and then lastly, I think this is where why I'm here now is that I think tissue engineers need to understand these rules and principles of morphogenesis in order to design novel tissues and materials for tissue regeneration. Okay, so we envision that there are really three roles for mechanics during embryogenesis, and we can think of them in the context of this sort of network diagram. As I came from physics originally and went into developmental biology fairly late in life, but I've come to love these sort of network, sort of gene network models. So I think you can begin to think about information at this level operating within multicellular tissues. You have, especially in the embryo, you have information provided maternally that's stored in the egg. It's carried from the egg all the way through early development. These factors and cues in the early egg contribute to patterning on the level of gene regulatory networks that control specifics of what genes are expressed in what tissues. And then you have growth factor signaling, which reiterates and feeds back to these gene regulatory networks. Now, cell identity is nothing if not to set up the morphogenetic program within those cells. So the key point of creating these patterns of different cell identities is to turn on the expression of morphogenetically active proteins, things that control the cytoskeleton, control the adhesion, control extracellular matrix, levels of factors like Rho GTPases that can control contractility and stiffness. And then these, when cells have these different arrays or sets of proteins expressed, they engage in different cell behaviors. These are macroscopic properties of cells, and whereas these are just sort of molecular expression profiles. Okay, so once the cell behaviors are initiated and triggered, these cells are capable of generating force and are capable of establishing the mechanical resistance or stiffness of a particular tissue. And then once you have these features established, you can then drive directly the shaping of tissues. Okay, so I think developmental biologists have been focusing on this particular role of mechanics in development for probably by now about 125 years. Okay, now these other two roles are really recent inventions. The idea that developmental programs must be robust in a way to accommodate changes in the microenvironment or changes in the variation in, say, maternal contributions and the operation of signal transduction networks. This now is thought to operate where bulk forces and stiffness might have some sort of crosstalk to one another, and may also operate on the level of these proteins directly, where one multi-protein component could talk to another one within the cell and adjust the properties of these structures like focal adhesions quite locally. And then lastly, I think tissue engineers and stem cell biology has been suggesting now that there are cues to guide cell fate embedded within the mechanical properties of the early developing embryo, and that these feed back not just to sort of cell behaviors, which we can see sometimes, but feed all the way back up to this really fundamental level of information, controlling and changing gene regulatory networks. Now, we've known that there are cues provided from tissue movement for a long time. Developmental biologists know about if you've heard of the term induction, and that term induction means that a tissue that has moved out of one microenvironment into a new microenvironment can now signal to create a third tissue type. This function of cell fate decision has been known about for a long time, but I think the ones where it depends critically on just the local microenvironment is relatively new. So we like to think about the embryo as a machine, and I'm going to have this equation is my major equation. So there may be one more complicated one later on, and I think you've had a lecture where you talk about stress and strain. This is essentially strain on the left-hand side. Strain is dependent on the amount of force applied to a material, and its effectiveness, the effectiveness of that force is determined by the mechanical resistance. So something that's stiff, if you push on it, it deforms very little. Something that's soft, you push on it, it deforms a lot. And so when we think about this equation, we can think about how it relates to the movements in the early embryo. So on the left-hand side, this is typically deformation. This is what we watch when we follow embryonic development. We can be very quantitative about it, or we can be very qualitative about it. We watch how these tissues change shape. We watch how they move and the rates of those movements. And the more sophisticated people are now beginning to measure more engineering terms, and they're describing engineering terms like strain and flow. So we're beginning to move into a more quantitative world. But with all of these aspects, we don't need to perturb embryos. All we have to do is set up a movie, watch them. We can use fluorescence. We can use just a visible light. But you don't need to perturb embryos just for this, just to measure deformation. However, if we want information about the force and the mechanical resistance, we actually have to go in to these embryonic tissues and actually perturb them. We have to pull a piece of tissue out to measure bulk forces or bulk elasticity. We have to poke at them to see if they can, how they respond both mechanically and through signaling to these applied forces. So I think that in order to make these kind of measurements, we cannot make these measurements in the absence of perturbation just by watching. You can't look at the gene expression profile and say that this tissue now has a stiffness of 1.5 kilopascal. We currently don't have that ability. Okay, so in trying to think about thinking about the embryo as a machine, we really want to understand how it works and then why it breaks. So here's an example of two embryos. On the left-hand side is an embryo that's developing normally from the process of just having finished gastrulation. And on the right-hand side of this is an embryo that's overexpressing a member of the planar cell polarity pathway, strabismus. Okay, so what you see on the left-hand side is the beautiful elongation of the embryo. The tail of the embryo, the head of the embryo are separating from one another. You have the rising of the folds, which enclose the central nervous system. Okay, so at the end of this movie, which is a few hours after the start, you have eyes forming and you have the tail. In this mutant embryo, this embryo is very defective. The head is right here, and instead of the tail moving further away, the tail is right next to the head. So I don't know if Ray described this particular kind of mutant before, but I like to think about it as a head aggregated to a butt, and you could call this a butt head mutation. All right. So this is a really lethal flaw in the development of the embryo. And we're particularly interested in just really what goes wrong under these conditions. Okay, so we come back to this, this basic equation. And so in order to study this further, I think that as engineers, we need to develop imaging tools to detect cell and tissue scale structures. We have to build and design structures, devices to control the microenvironment, and some of these are going to be mechanical, some of these are microfluidic. I would say I'm interested in the question, and I'm eager to work with people to build whatever kind of tools and devices that we need to make these kind of measurements. And so that's led us into a number of different collaborations, and I'm really proud of being able to work with people who are more engineering focused than we are on that. And then finally, there's a major problem coming up on the horizon, and that is with the high density imaging information that we're gathering. Single experiments can collect a terabyte of imaging data. How do you go about analyzing that? Well, you could start with maybe 100,000 undergraduates working away, clicking and hand digitizing. But I think you're going to quickly run out of undergraduates that way. There are data processing tools that are being developed that will help us understand these. Okay, all right. So the focus of the rest of the talk is on the process of gastrulation. And Ray maybe told you that the blastula develops into the tadpole, and this is essentially a transformation from a ball of cells into a hollow tube of cells. This is a topological transformation, first into something that looks from a ball of cells into something that maybe looks more like a bagel. And then once you have the bagel formed, the bagel elongates into a nice long sort of pasta-like shape, like a penne, right? So you have this early development which involves large-scale movements. of endoderm, mesoderm, and ectoderm, and places those tissues into their proper location. This stage is something that you can think about as your house. That's framed, it has the roof, it has all the basic structure, but it doesn't have any of the organs yet. There's relatively few neurons, there's no functioning gut, there's no organs branching off the gut. This is a sort of a blank tablet for the establishment of organs and limbs and everything else. Okay, so our model system that we're focusing on is the Xenopus embryo, and this is the process of gastrulation and neurulation that I was just showing you. But the frog embryo has tremendous advantages to my group as engineers, because we can take embryos out of they're fertilized in vitro, so we don't have to worry about culture conditions very much. They're relatively large in size. Development is fast compared to mouse, which takes days to gastrulate, or even in embryoid bodies, it takes days to reach that stage. These reach that stage in less than 24 hours. And in the process from 24 to about 34 hours, they've completed the basic construction of the house. They've moved all their tissues inside. There are very simple culture conditions. We culture embryos on the bench top at room temperature in defined salt solutions. So as engineers who always seem to have problems keeping their cells alive or working with sterile technique, these guys are so easy to work with. So cheap and fast, I'd have to say. Okay, so there's good news and bad news about studying early development. The bad news is that Xenopus gastrulation is deceptively simple. You can watch these movies and get a sense of what's going on. The folding, the deformation. However, they're accompanied by a series of complex internal movements. Sort of in this region, there are a whole slew of internal movements within the embryo and that involve multiple cell types, both mesenchymal cells and epithelial cells. The good news is that there's been 100 years of experimental embryology where each one of these particular processes can be tested in a specific tissue explant. So you can cut the embryo apart and test each one of these phenomena within an isolated tissue fragment. Okay, so that's the good news, and that's the result of what's called the cut and paste embryology of amphibian embryos. Okay, and one such example of cut and paste is used to understand the process of convergence and extension. So Ray told you all about that, I'm sure. Just to sort of highlight this again, if you cut and paste a piece of tissue and take it out of the embryo, all of the processes that are operating within the embryo are recapitulated in these tissue isolates. So just I think like the processes operating with an embryoid body, as you can dissect it further, operate autonomously within this tissue. And so by watching this, we can see that this tissue self-assembles and autonomously deforms at a strain rate of about 10 to 20% per hour. And this is where the speed aspect comes into play. I won't cover this too much, but just to say that this process is driven by convergent extension, where a group of cells, so say maybe 2 by 2 cells, undergo a process of directed cell intercalation, which drives the elongation of this array of cells, so that the sort of head cell here is pushed away from the butt cell at the posterior end of this array. So we come back to this equation again and want to understand what the mechanical context is that allows these cells to intercalate between one another to push the head away from the butt. And so the first thing we wanted to do was understand what the mechanical resistance of this array of cells was to any applied force. And so we started in my group adopting a device that was first developed in Ray's group by Steve Moore and Mimi Cole called the histo-wiggler. Now you really can't go about talking about the histo-wiggler in public too much. It affects your credibility somewhat. So, of course, as proper engineers, we created a 3-letter acronym, the Nano-Newton Force Measurement Device, or a 4-letter acronym. This is a device that allows us to apply an unconstrained uniaxial compression test. The way that this works is that we put a piece of tissue on this apparatus, and this is one of these pieces of tissue where that's been excised from the embryo. Say the head, it would be at this end and the tail end would be at this end, except we've cut them off, and then we can compress them between parallel plates. We can measure the force of resistance of this block of tissue, the amount of strain that's being applied, the cross-sectional area over time, and then we can calculate from that all the mechanical parameters that describe the behavior of a standard linear solid material. So we can look at this piece of tissue and pull out parameters of elasticity and its viscous behavior. Okay, so I'm not going to go through all of the experiments that we've done, but just sort of try to summarize the conclusions of this basic descriptive work. So the first thing that we discovered was that, of course, if you look at this elastic response, you realize that tissues are neither liquid nor solid, but really viscoelastic. The surprising thing to us was that when we look at how much strain that goes on, how much deformation occurs over this short period of time, very little of that strain is stored in the embryo permanently. So at any time, the embryo has about 5 to 10% strain that it's storing. And that's about 20 to 30 minutes of deformation. After that, those strain energies are dissipated by the viscous nature. Okay? One other surprising thing that we found was that dorsal tissues, these tissues stiffen dramatically over 8 hours. So in that time course of the movie that I was showing you, the embryo stiffens 6-fold. So it's like you woke up this morning and you weighed 100 pounds. You're great. You wake up, you go run 6 miles, you come back. But throughout the day, you're getting stiffer and stiffer and stiffer. And by the end of the day, you weigh 600 pounds. And I would dare you to go out and try to run another 6 miles. But the embryo seems to do this without effort. The rates of elongation, the rates of tissue movement and deformation are linear throughout this whole time course. In watching that movie, you didn't see things sort of slow down as the process proceeds. Now, the tissue stiffnesses that we're talking about here, though, are ultra low. If you look at the actual units here, the embryos start off at less than 20 Pascal. And when I say they stiffen over time, they're going up to maybe 100 Pascal in stiffness. These are ultra soft tissues. Okay? And to compare them to the mechanical properties of adult tissues, you see very quickly that there are very few adult tissues that are down in the level of stiffness of these early embryonic tissues. But I think stem cells are one of the places where you see a very similar transition from early soft to later stiff. Now these measurements are not extendable to every type of embryo. In fact, as a grad student, I measured the mechanical properties of sea urchin embryos at the same stage, and they're in the kilopascal range. So this is not extendable. I think we have to understand each embryo in the context. If we're going to do comparative embryology, we have to go and measure mechanics in those embryos. As we were doing more experiments, my grad student who is doing this took on a heroic series of microsurgical manipulations where we could cut various pieces of that tissue out, make new explants that are really impossible to make genetically, and test the mechanical properties of those. And so he made a series of very complex explants, but found ultimately that there's a remarkable variation or diversity in mechanical microenvironments at the same stage. So endoderm tissues here, and shown in yellow, are very, very soft. They're much lower in stiffness than the bulk tissue. The mesoderm here, the cells that are undergoing convergence and extension, are actually 9 times stiffer than those endoderm cells. And And lastly, I think one thing that we didn't realize or really sort of fail to realize on a regular basis is that there's tremendous amount of embryo-to-embryo variation in stiffness and even force production. We see a twofold variation between siblings. So you can look at a dish at 100 embryos and expect to see somewhere in that dish, invisible to you at the moment, that some of them are very soft and some of them are twice as stiff as those other embryos. And yet they all develop at the same rate, and they develop and reach maturity. Yes?
How about the ratio of the endoderm to, for example, soma?
So we haven't been able to do that. So these set of microsurgical manipulations do show quite a bit of variability in the stiffness of those tissues, but there's a lot of variability in these microsurgical manipulations. And I'd say I could jump to say yes, we see variation. We do see variation, but I'm not sure that can't be attributed to the microsurgical manipulation. So once you start cutting into an embryo, things become a lot more difficult to that sort of question is hard to answer. Okay, so as a cell and developmental biologist, we started to ask the question of how stiffness was controlled molecularly. And we could have gone and done a microarray and come up with the top 25 candidates, but instead we took out a piece of paper and just wrote down some of our favorite morphogenetically active proteins. And so top on the list was a set of extracellular matrix proteins that are expressed within the embryo at these early stages that increase in expression over time. And then we listed elements of the cytoskeleton, actomyosin and microtubules, as critical candidates. And we have also all sorts of other things that we've not yet tested directly. But just to give you a jump on this, we were able to rule out the role of the extracellular matrix. We could knock down extracellular matrix synthesis, make measurements of stiffness, found absolutely no change in the stiffness. This was most disappointing to me because I'd done a whole lot of work as a postdoc on fibronectin synthesis, fibronectin scaffold assembly, handling and manipulation of extracellular matrix. But you wake up the next morning and you say, okay, let's just keep going. It's good to know. I mean, I think this is remarkable, but it's not too unexpected because the embryo doesn't really start to express very stiff extracellular matrices like collagen until several hours after these measurements were made. What we found instead was that actomyosin was the critical contributor of tissue stiffness in these systems. Even microtubules, we could knock down microtubules, and surprisingly we found when we knocked down microtubules, we found the embryo stiffened. But that was due to a rather totally a biological issue in that there are activators of Rho GTPase that are bound to microtubules. And when you depolymerize microtubules, you free these actomyosin activating proteins. And once we excluded those proteins from the embryo, we found that there was absolutely no change in stiffness when we knocked out microtubules. Okay, so we have this set of questions where we now understand what the subcellular proteins that are responsible for mechanics, and we understand their effect on the bulk force mechanics. So we realize that subcellular actomyosin dynamics are effectors. We can now show that we can actually do experimental biomechanics on these tissues, even though they're very, very small, and that we can describe the material properties of these tissues quantitatively. Okay. So we wanted to start asking more fundamental questions about how these tissues changed with time and how programs of feedback were actually operating. And so what kind of mechanosensing and feedback might there be? So we have this sort of top-down array, and we started to think about how sensing might operate. So on one scale, the very, very fast scale where there might be multi-protein complexes such as myosin or complexes within the focal adhesions operating. The ones that were particularly interesting to us were these sort of moderate scale feedback mechanisms. All of these are cell autonomous. And so when a cell feels a mechanically stiff environment, it can compensate. But how does a tissue do that? How does one cell over here compensate for mechanical events that might be happening 10 or 100 cells away, which we see in the real embryo? We also see slow scale events, maybe operating on the life cycle of the embryo. These sort of feedback or sensing approaches might occur based on the maternal contribution to the egg or on the influence of environmental factors. And then there are, of course, very, very slow processes, generation. This is evolution. Sorry, I have to include evolution because the process of evolution is ultimately what controls the design, the initial design of these programs. So we started with a set of really puzzling observations from the early embryo. And one of these was that certainly that embryos will elongate at a constant rate even though their stiffness is increasing. Embryos with moderately reduced stiffness, and these are sort of across the board. Any treatment that we make that moderately reduces the stiffness of an embryo leaves its development unperturbed. But any treatment that we apply that increases the stiffness of that embryo, sort of 50% or doubles the stiffness, we see those embryos develop very defectively. So softening seems to be okay. Stiffening seems to put those embryos at risk of lethal phenotypes. Okay, so to understand this, these puzzling observations, we sort of return to this equation. So one side, deformation, we know that when deformation goes wrong, we have these defects. We know the mechanical resistance is being decreased. So if you decrease mechanical resistance, then in order to get the same rates of deformation, maybe the embryo is accommodating by decreasing the force production. Yes?
Actually, you said that gastrulation is like a linear process. So do we able to understand that at any point of time there is a maximum force and maximum resistance?
That's a very good question. So you're jumping ahead of me just a second. In my next button press, I was going to circle this aspect force. So I've only now just told you about measuring mechanical resistance, and the question is whether we can know what the forces that are being generated by these tissues. So a very good question, and it's the next part of the talk. Okay, so we needed to measure force in order to get to these questions. And so we started with an approach that was developed in Ray's lab by Steve Moore to measure the force of elongation of a single explant. The problem with this approach is that these embryonic tissue explants are kind of like really soft bars of soap. If you try to grab them in any way, you try to compress them, they slip out of your hands, or in this case, the grips of the apparatus very, very quickly. So rates of error and drift in these particular experiments are very, very high. Although we can make individual measurements, we can't do so in a systematic way with high enough throughput that we can do real tests. So what my student Jianzhou did was to develop an approach using force reporting gels. So you've probably heard a lot about polyacrylamide gels that are used to measure the traction forces of individual cells. We took sort of a slightly different approach and a very different scale. We cut out our particular tissues and embedded them within an elastic gel, and then solidified that gel around those tissue explants, and as they elongate, they push on the walls of this gel, their cage basically, and we can visualize the deformation and strain within these elastic gels. And so taking that information, we confocal both the explant and the gel itself with a bunch of fluorescent beads embedded in the gel. We can calculate displacement maps in that gel and extract out the stress profile that's produced by an individual explant within a gel. Now we can do this very nicely in a relatively high throughput way, so that we can embed, like 20 explants in a gel. As long as they're separated from one another, we can put them on a stage of a multi-position controlled confocal microscope and make a lot of great measurements simultaneously. And so I'll just sort of take a look at these sort of control experiments, but you have to validate methods for measuring gel deformation. You have to test the accuracy of this method. So we used standard Hertzian contact methods to validate this. All of this stuff, of course, goes in the appendix of individual papers, because it's not really part of the paper. It's just part of the certification process, you might think. And then we took tissues that don't actually extend and saw just that they didn't actually generate any stress. So then we started to map out the 3D profiles of stress production. We can't look through the entire tissue explant because it's opaque. Lasers don't go through opaque tissues, frog tissues, very well at all. But we can sort of fake these 3D maps, not really fake them, but just by reorienting the tissue explant in the gel, we can look at the force profiles or the stress profiles surrounding these differently oriented explants. And so most of the stress is produced along the anterior-posterior axis of the explants, a little bit in the dorsal ventral as these tissues try to thicken along with extend. So to get to this question of elongation, we wanted to know whether tissues could adjust their production of force in response to different mechanical environments. We can't, yes?
Given the resistance of the gel, would the deformation of the tissue be the same?
No, that's right. So the gel is elastic, right? So it cannot extend indefinitely. So what happens is that the tissue extends to a point where it comes into mechanical equilibrium with the gel. And so we see them extend, and there's sort of a plateau in the amount of stress that's stored in the gel. So yeah, the explants themselves don't continue to elongate. However, because the inside of this gel is non-adhesive. So eventually, the tissue will try and extend and extend and extend, and ultimately it buckles. It basically enters Euler buckling and flips within these gel cages. And if you follow them for many hours afterwards, you see that they do flip. We can't use those measurements because at that point they've buckled, and you're not measuring the. Oh yeah, we see force that, where if like I bend like this, then you would see stress at the point of buckling as well. So yeah, you can get the measurements, but they're not so easy to interpret. Okay, so just like you would with traditional polyacrylamide gel approaches, we put these tissue explants in increasingly stiff gels and then measured their ability to generate force or stress. And what we found was that with very low stiffness gels, they produced maybe 3 to 4 Pascal of stress at their maximum. But when we increased the stiffness of the gel, they started to produce more and more stress. Now, they don't compensate fully for the stiffness of their surrounding microenvironment. They cannot generate enough force against a 500 Pascal gel in order to keep their rates of deformation the same. So what we think is that they're really plateauing here in their ability to generate stress. So this might be an indication of both their ability to compensate for some amount of increased stiffness in the early embryo, but might account also for the defects that we see when we increase stiffness just too much. Okay, so we wanted to ask what tissue generates force. If we look at anatomically those tissues within gels, we can see what tissues look like when they aren't in gels, and we can see what they look like when they're in these super, super stiff gels. And we've seen notochord development that looks, here's a case where you might actually see a tissue buckling within an embedded tissue. So this piece of tissue is now basically showing that it may be buckling. And so we thought first that the notochord was generating this stiffness, but again, through the process of microsurgical manipulations, we showed, in fact, the notochord has absolutely no contribution to that increase in stress production. So we know a lot about the mechanics of the early embryo at this point. We know actomyosin is a contributor to both stiffness and force production in experiments I didn't show you, variation, and the sort of basic description of mechanics. But if we want to know about more complex problems, we're sort of left. We're trying to understand how feedback works and at what level. But with these complex systems of embryonic development where you have multiple cell types, multiple tissues, it becomes very difficult to answer these. So we stepped back for a little bit to look at to sort of list the challenges of measuring the mechanical movement, physical mechanics of endogenous movement. So in one regard, we can't control the timing or triggering of morphogenetic events. No one I know in developmental biology has a clue about what really triggers morphogenetic movements. They think that it's just the timing of gene expression. But in many cases, you have movements that occur simultaneously across many cell types. And it's thought that there's some local community effect of communication. As I described already, tissue architecture is constantly changing in the early embryo. This dorsal axis elongation is one example where we think that the tissue architecture does not change that much, and it makes it possible for us to look at this. But if you want to understand how the heart develops, for instance, very complicated changes in the anatomy go on consistently with the force production. And then it's difficult to measure force production using these varying complex morphologies and geometries. So one way that we could have gone is to start making very complex computer simulations of these complex geometries and complex force profiles. But we decided to go the opposite direction. And I think we wanted to follow the approach that skeletal muscle physiologists have taken, and they were faced with many of the same problems in understanding muscle function in the early 20th century, and so they approached it to first be able to induce contraction ectopically from, say, neuronal control. They built devices to maintain and hold constant muscle architecture and measure force production. And so we turned to a different part of the embryo at a different stage to start to measure this. We turned to the embryonic epithelium. So the epithelium is mechanically accessible. It has all the same advantages of the rest of the embryo, large cells and a fate map. But we can access this tissue at any stage, and we can visualize the actomyosin dynamics within that tissue. And so what we decided to do was build an aspiration-based approach. Instead of microaspiration, where you can measure the mechanical properties of sort of parts of a single cell, we built a gigantic aspirator, one that is 150 microns in diameter, 125 microns in diameter. Bring it up to the wall of the embryo and suck a small patch of tissue into the embryo. And furthermore, what we discovered was that we could apply an electrical pulse to stimulate contractility in that epithelial sheet. So if you take a line and draw it along this view of this tissue that's being sucked into this microchannel and project it over time, you create this chymograph. So here's the embryo at rest. We decrease the pressure inside the microchannel. It draws part of that tissue in. We stimulate with an electrical stimulation. We can see an autonomously produced contraction in that array of cells. And then spontaneously it stops contracting, and then the embryo continues to move in. And then we can decrease the pressure in the microchannel and see relaxation of that tissue at the end. So we can calculate from this, we can calculate the viscoelastic material properties and the force generation capacities of this piece of tissue. And we've since gone on to show that the force generation capacities of these tissues are analogous in both the timing and the performance as the actomyosin contractility of individual cells. So it has the same sort of time constants, involves many of the same proteins that activate. We can block this with myosin function inhibitors. So now I think we have some approach where we can control the precise timing, we can control the architecture, and we can make force production measurements within this easily defined architecture. So instead of turning complex morphology and geometry into simulation, we turn the embryo into a very simple model of a material property that's electrically stimulatable and produces morphogenetic force. So we first wanted to see whether all of this was linear. I won't cover that. But one of the questions that has arisen in development is whether tissues are mechanically sensitive. So if you poke an embryo or you somehow drive mechanical resistance, put an embryo in a more resistant environment, will it increase or change its own stiffness? And so what my postdoc did was to test a number of different, a large number of embryos with this electrical stimulation to see if application of force resulted in a change in the stiffness properties of those tissues afterwards. To do so, he developed he applied some very simple power law models of viscoelasticity and calculated this beta constant. And this beta constant in our tissues, about 0.3, is almost exactly what you see in actin gels and in even single cells. So he also wanted to know whether the rate of strain would also change the mechanical properties. So there's a lot of groups that are postulating that embryonic development depends on and can sense the rate of strain and can accommodate mechanical strains in some novel way. And so he applied different ramps of stimulation, mechanical stimulation, and could then measure and fit with his relatively simple mechanical model the responses of these tissues to these different changes, these changes in ramp, and found that the power law model representations, the beta does not change at all over the time course of these. So these tissues they're not undergoing fluidization with high strain rates, and they're not stiffening or changing their mechanical properties over time with the application of strain. However, some of the interesting things that he did find was that stiffer tissues in the embryo, ones that are at the high end of stiffness, produce stronger contractions than tissues that are actually soft. So he found a very strong correlation between stiff and strong contractions. So this could be an indicator of one of these mechanically sensitive programs of development. Okay, so in some sense, we've somewhat rewritten this equation of deformation, force, and resistance into a more classical closed form integral equation that relates strain to compliance and the change in stress. And we're working with this very simple linear model, closed form linear model of which we think of as almost like a model of morphogenesis to explain morphogenetic events in the embryo. Okay, so I think we're beginning to uncover the molecular basis of these processes, and I just wanted to finish up with just a couple of slides on some work that was carried out by a very talented grad student who was collaborating with our group. Actually, he's sitting right over there, Tony Kim. He's now doing a postdoc in the Langer lab. But he was working with Phil LeDuc, who's at Carnegie Mellon University, which is probably a stone's throw away from where we are, University of Pittsburgh. And Phil and I have been talking about collaborations, and Tony personified those theoretical talks between PIs and actually got some really interesting work done. So Tony started to develop microfluidic arrays, devices that could apply very precisely streams of chemical components to the surface of tissue explants and look at how those tissues respond to those stimuli. So to really get an idea of what happens when you stimulate a tissue and what happens in the response of that tissue. We've done a number of different different things. Some of them are already published, some of them are in review right now. And so with this approach, we can probe multicellular dynamics with direct chemical stimulation, both long-term and high-speed control of timing. Tony built devices such as this to control those long-term streams, and we could precisely stimulate even a stripe of cells that's, say, 1 cell diameter across within a complex tissue. So I think this opens up for us a range of opportunities to control gene expression, because you can control, if you can control the microenvironment, you can control the spatial organization of gene expression within tissues. You can control the signaling within tissues with inhibitors and activators of signaling systems. And I think this is great potential for exploring in a 2D system the responses of these tissues to these strains, to these factors. So to summarize, I think we've gone through the roles for mechanics in early development, and I've shown you some ways that we have of measuring those mechanical properties, measuring viscoelasticity with this sort of stress relaxation test or microaspiration. We can measure force in these tissues, traditionally with polyacrylamide gels. We've done that with tissue explants. We can also measure force production, bulk force production, with these gels. But I think there's a whole range of major questions that remain. One of these are, some of these are, what are the potential mechanical stimuli within tissues? We have a lot of hypothesis out there about the role of mechanics in morphogenesis. Very few of those hypotheses have been directly tested. I think it's really critical to develop experimental techniques to go in and probe and actually test the role of mechanical stimuli during development. So we need to know whether cells really sense these stimuli. Do they really sense them, and can we visualize their changes on a time scale that is useful for us? So if you have to rely on tissues to undergo changes in cell identity to assess their response to a mechanical stimulation, I think that's too long. You need to see what happens immediately, what signaling pathways are being stimulated, what events within the cytoskeleton are being changed within minutes, not hours or days. With that kind of information, I think we'll really be able to make some headway on understanding the molecular mechanisms, what the responses of the cells are, and then lastly, whether we can control the stimuli with the expectation of a particular response. We want to be able to routinely know how we're stimulating a cell and predict what the response of those cells are to that stimulation. In order to build these sort of engineering modules, you need to know these things. You can't just say, there's a big black box. I think we need to know how those cells are responding if we want to be able to control them within a complex multicellular environment. And with that, I'd like to thank, first of all, Ray Keller and my postdoc mentors a number of years ago for bringing me a biophysicist into their lab with no wet lab experience whatsoever. I had no idea how to operate a pipetter, a pipetteman. I didn't know what the yellow tip was versus a blue tip. And then also to thank the members of the lab who worked on a lot of this, did a lot of this work. Mickey van Dassau, who came to me as an integrative biologist who was interested in ecology, development, and evolution. Three of those things, two of those things he worked on in my lab, so ecology and development, and now he's back at near an ocean at the Duke Marine Station doing more of that. One of the students that worked on the actin dynamics was Hae Young Kim. She's a postdoc at Princeton. Tony is here at MIT. Jian Zhou is working at UC Irvine as a postdoc in Stephen George's lab on tissue engineering of cardiac replacement tissues. And I thank all the collaborators that we've had, specifically Phil LeDuc and Bill Messner, who've been instrumental in helping us formulate these sort of control theory ideas of how embryonic tissues are stimulated and respond to those stimuli. So thank you very much. Stunned.
We have time for some questions.
Oh, good, good. Yeah?
So I have a question. Just like you were showing the migration of the lateral line cells,
right,
zebrafish, across like a well-defined path. So is there any contribution from the ECM that guides the cell, and what are the factors that actually coordinates the? So I would have to say that I think there are very few what are soluble factors. So keep on talking about soluble factors. As engineers, we love soluble factors because we can control them. I think in embryos, nothing is soluble, right? The activity of proteins are always felt very close to where they're synthesized. So in that particular case, there's a protein SDF1 alpha, which is a chemokine that is secreted and assembled into an extracellular matrix that actually runs along the entire length of the zebrafish embryo before those cells even arrive. So the embryo has already made, in that case, a roadway for those cells. They have CXCR4, CXCR7 receptors for SDF1, and they are primed to basically sense that roadway, gobble up, actually, the SDF1 alpha that then drives the polarity of cells within that slug. So they don't. So because they have this stripe of signal to track on, they could just walk in one direction in this stripe and then sort of get distracted for a minute, turn around, and walk back on the same stripe if they didn't have some persistence in polarity. And the sort of the construction of that slug allows them to develop a polarity within it that gives them the ability to use the permissive signal of the SDF1 in an instructive fashion. So, yeah. What actually prevents the cells like from going back? Because we see that at the final state, actually we'll have a full track of cells deposited in the state.
What keeps them from going back? So the receptor CXCR4 is expressed at the leading edge of that slug, and the receptor CXCR7 is expressed at the back half of the bus, basically. So CXCR7 only works to sort of sponge up the SDF1 alpha and doesn't signal. SDF1 alpha does signal through CXCR4. So you have the polarity built within this slug, where at the rear of the slug, cells don't sense it. All right, all they do is they follow along with the cells that are moving. The cells in the front are the ones that sense it, and they're the only ones. So the polarity, the sort of, what I was talking about, the feedback loops, in that case, are operating
within a few hundred cells within that slug that give them the polarity to migrate in what we would consider like a road, an open road. But if you think about it like driving out on the street, I don't know, here Boston might be a special exception, but in Pittsburgh, people drive on the right-hand side of the street, and they all go in the same direction. So they follow. The roads aren't meant for cars to be going in both directions, opposed to one another simultaneously. There are cues for drivers that polarize their movements. Cars generally don't go equally well in the back direction as the front, even though they can. So I think if you take, just have to start thinking about how these modules are designed. You can start to see how they actually control directed migration versus just, why is it that those cells just don't spread out on the stripe and you get a constantly, dense array of cells along that SDF1 alpha stripe? But yeah, I mean, those are key questions. Those principles of directed cell migration are just now being worked out. I mean, there's exciting work going on now that's figuring out those principles. So I think we're at the right time as tissue engineers to start reading that literature very, very closely, trying to pull out key features and say, well, okay, if we're going to build a tissue, we have to know how to connect a small cluster of cells over here to another cluster of cells over here. And we have to do this in a dense tissue, right? So if you want to make that, you want to make those connections, those wiring connections, you have to be able to drive that event. And how would you go about doing that? Substrate patterning, microfluidics, optogenetic controls, all of those processes are available to us, but they're not necessarily operating in the embryo. Yep.
I recently showed a video about so you cut and paste tissue and observe this similar convergent extension. But I noticed that it seems to me the elongation there is more significant. Is there any particular reason?
Okay, so one of the reasons why elongation is so different when you take them out and put them on in a dish is that we typically, these cells are expecting to see a fibronectin substrate. And when we take them out of the embryo, we give them a fibronectin substrate, but it's attached to glass. That's now a novel mechanical microenvironment for these cells, where normally they would extend attached to the substrate or attached to their neighbors and contract. Now they're attaching to their neighbors, but their neighbors are glued to glass. The substrate that they're on is glued to glass. When they try to contract, they can't. They can't overcome the mechanical resistance of the glass, and they get very, very long. So the aspect ratios of cells as they intercalate on glass can be like 6:1, whereas if you look in the embryo, they're never much more than about 2 or 3:1. So that's the issue, is that the material properties of the embryo are viscoelastic. They're not elastic, right? So that, as a cell grabs its substrate and pulls, there's viscous dissipation as that cell contracts. So you're not storing elastic energy in their substrates, which are other cells. So I think one thing that came out of this talk was some exciting work by Dave Mooney, who's building viscoelastic substrates for cell migration. Very exciting to me, because I think viscoelastic substrates are much more like what embryonic tissues are, not elastic. Yeah. Yeah.
So for the elongation of the tissue showed embedded gel, maybe I missed. What is the mechanism for the elongation? Is it like realignment or?
So those tissues are elongating because of individual cell rearrangement within those tissues. So the process of convergent extension is operating within those tissues. So individual cells are trying to intercalate between one another, and they're pushing the tissue to elongate. And because it's trapped within a gel, it generates force in the embryo, but trapped within the gel, it pushes against the elastic cage of the gel. And so that's really what we're measuring. We're not measuring the actual force in the tissue. We're measuring the reaction force that's needed to constrain that tissue within that cage. And so our gels, of course, are elastic, and they don't viscoelastically deform, or they're a little bit viscoelastic, but on this scale they're elastic.
I realize, so this is after the surgery, so it's a separate tissue. If they
Right.
are in the in vivo system, they actually have to also migrate relative to the other tissue.
So,
Mixed,
yeah, so this tissue isn't doing much migration. Most of the rearrangements are local. It's not really understood how much shear. So you're sort of asking about tissue shear. There's a little bit of tissue shear within this tissue, but not a whole lot. There are other events during gastrulation and later during morphogenesis when tissues actively do slide over one another great distances, but in this particular case, not so much. Yeah.
Hi, do you have actually two-headed frogs in your lab? I mean, you do microsurgery and put one so do you happen to actually develop into a full-grown frog with two heads?
Yes, you can make frogs with two heads quite easily. There was an experiment done by in the early 1900s where Hilda Mangold, working in Hans Spamann's lab, grafted a piece of tissue from the prospective head region of the embryo into another embryo in the prospective I'd say posterior region. And what those embryos develop are two heads on the same embryo. And really the remarkable thing is that those heads are connected. The two heads are at one end, and they share one tail. So they join in mid-body. So they have two notochords, two eyes, two heads. I don't know. They never, I don't think, they don't do well when they reach more advanced stages. But you see, you check on the internet, conjoined twins, and you can find all sorts of animals have conjoined twins. The remarkable thing is if you look at all of those animals, all of those examples from the internet, and you'll see that they always have that's always duplications of the anterior. It's never duplications of the posterior. So they may have two heads, four arms, but they'll have two legs, or they'll have the regular array of legs. So the process of gastrulation starts first anteriorly and then goes posteriorly. In the frog embryo, the mouse embryo, the chick embryo, they all work the same way. And if you put two organizers in that will induce two heads, those processes will join together but share a common tail. So how the mechanics of that works, how the signaling accommodates that to produce something that even looks recognizable, I think is very remarkable and tells us a lot about the robustness. I have a couple of movies I could show you later, but working in a lab, a developmental biology lab, everyone appreciates kind of freaks of nature. So, if you see something weird happening in your dish, you call over everybody in the lab. It's like, ooh, look at that. And there's two heads forming, or there's an embryo with two blastopores. You just set it up and make a time-lapse movie and take a look at it. But I think there's some remarkable aspects of robustness that are we can also see where there are apparent defects in early development, but the embryos accommodate those and develop into perfectly normal tadpoles. So on one hand, it's really important for genetic analysis of the signaling and gene regulatory networks that development is fragile, because you have to be able to break, pull proteins out and see what kind of phenotypes they make. But there's other aspects in which you can perturb, you can pull some proteins out and you see no defects whatsoever. Geneticists and developmental biologists have terms for that. They call it redundancy. Whenever you hear the word redundancy or penetrance or something like that, it means they don't have a clue about what's going on. Okay? I hope that's not recorded. But, all right. Yes?
You showed very simplified model. So what if you consider that shape or geometry, and is it a completely different output, or if you consider biaxial force instead of the uniaxial?
So you're asking if we change the geometry of the compression tests. So we do the stress. Yeah.
Complex geometry.
Yeah. So I think I understand. So one of the reasons why we chose that specific test and the specific tissue is that for a stress relaxation test, a uniaxial compression test, you rely on the material to have the same cross-section and the same composition along the length of that beam or column. I mean, if you talk about, applying this to an I-beam, for instance, you would only apply it to the ends of the I-beam. It wouldn't make any sense to apply it to the sides of an I-beam, right? So the same is true in the embryo. These tissues are complex in their sort of laminar structure, in their cross-section, but that cross-sectional structure is very similar from the posterior end of the embryo up to the anterior end, at least in the pieces that we're taking. So we're reasonably confident that we can interpret the experiment. So if you do the other experiment where you compress it in the other direction, where very complex morphology, very complex anatomy, I don't know how to interpret the results. Right, it's like taking an I-beam and compressing it in the, across the structure. It's not easy to interpret the results. You would need complicated finite element models that would inherently require the information that you're getting from the from the axial compression tests. So it would be really just a validation with these other geometries. So. That's why we turn to the microaspirator, which is a more isotropic measure of mechanical properties. And we can do this at a time in the embryo's development where there's relatively little anatomy that we have to account for. So we could perturb the molecular composition of those tissues and not have to rely on the embryo to build this axis for us to test in the uniaxial compression device. So that's actually a great utility of microaspiration. We don't require a correct geometry to start with. Yeah. Did I answer your question? Thanks. Yes.
I was wondering for the relaxation test that you mentioned, if you consider the linear model you showed, if you tried using simple non-linear empirical functions like the state exponential epithelium.
Yeah, we could use any kind of constitutive model for viscoelasticity and get pretty reasonable fits. So we chose the standard linear solid model because it's relatively easy to look at the graph and extract out your K1, your K2, your residual stiffness versus your instantaneous stiffness, and the viscous response. It's just really, it's easy. We could get into a complicated hours-long discussion about which constitutive material model to use, but I think in this particular case, we didn't rely on the model to do our experimental tests. We looked at the stiffness at 180 seconds for all of our comparisons. I don't like to calculate something and then compare it experimentally. I like to have something that we observe and do those comparisons, just to take a little bit more out of the mix of variability and interpretation. So that did change a little bit when we switched to using the microaspirator to measure stiffness. And there we did test a number of different models, constitutive models and structural models. We looked at surface tension as a